Thank you. All right, we're going to move on to our next presenter, Anna Kase. If you'll remember from earlier when I announced our award winners for this year, Anna was our PhD Student of the Year. Anna is a fourth-year PhD candidate studying under Dr. Jake Kerby at the University of South Dakota. Rocco, can you unshare your presentation, please? Thank you. Anna has worked extensively with turtles on the Missouri River in South Dakota, and her dissertation focuses on how human modification to the landscape impacts turtle distribution, abundance, and health. All right, Anna, go ahead.
Sorry, I couldn't figure out how to unmute myself while I was sharing my screen. I'm so sorry.
Oh, well, go ahead and share it and then I will unmute you. How's that?
Okay, I think I got it now.
Okay.
So sorry. All right, start. Okay. Hi everyone. I'm so excited to get to share our results from a really cool experiment that we got to do this past summer, looking at a predator-prey interaction between a threatened turtle and an invasive bivalve species here in South Dakota. So the invasive bivalve that we're talking about is the zebra mussel. So there are a few attributes about zebra mussel biology that really that have allowed them to become global invaders, where they can be found throughout many water bodies in Europe and in Central and Eastern North America now. So first of all, they're a generalist species. So pretty much if it's wet, they can survive there. There's no specific water quality requirements for them to be able to establish and breed. Additionally, they're filter feeders. So again, they're not specific in what they need to eat. Anything that's floating through the water, they're more than happy to pick up and assimilate any available nutrients into their tissues to help them grow and breed, which is the next point, is that they are prolific breeders. A single female zebra mussel can actually produce 1 million eggs over the course of 1 year. Those eggs are spewed out into the environment, and then they hatch into free-swimming larvae called veligers. These veligers are microscopic and can be carried in water currents or be sucked up into boat engines or ballast water and transported downstream to different areas in the same water body, or can even make jumps from water body to water body if they're in a boat. Finally, the zebra mussels can actually survive out of water, which makes them difficult to dispatch through desiccation. In completely dry conditions, they can close up and survive for about 5 days with the sun beating down on them. Otherwise, if they have established somewhere in a more moist environment that's a little more enclosed, like part of a boat engine or in an intake pipe where they don't have to be totally submerged, but the conditions are still moist, they can survive for an additional 21 days before they need to be resubmerged. So the zebra mussel invasion began in the United States in the 1980s, and this graphic shows how they have extended outwards from the Great Lakes through the major river systems in the United States. They are also found throughout different streams and lakes and pretty much anywhere that they could get into. You can see how they've started to move up the Missouri River in past years. They began that invasion in the early 2000s. And in South Dakota, began their invasion in 2015 in the Missouri River, where they were found in the main stem reservoir, Lewis and Clark Lake, and subsequently in the 59-mile stretch directly downstream of that reservoir. They stayed contained there, at least in the Missouri River, for about 4 years, until recently in 2019, Game Fish Parks officials found zebra mussels up in Lake Sharp. This is particularly concerning because this could potentially compromise Lake Francis Kase and the 39-mile stretch as well. Again, because of that free-swimming veliger stage, the larvae could go through the dam and begin colonizing downstream. So different impacts that the zebra mussels have, they grow everywhere. Any hard surface that they can attach to using their byssal threads. So they can clog pipes, they can cause damage to boats that are left in the water year round, and they can even begin encrusting native species like this mussel here, which can ultimately suffocate the native species that they have begun growing on. Other ecological effects, again, including casing large hard-shelled organisms. Beyond mussels, they've also been shown to encrust crayfish and have been found growing on turtle shells as well. They can also outcompete other filter feeders. So this competition could lead to differences and changes to energy pathways and overall food web structure after an invasion. And they can also facilitate biomagnification and bioaccumulation of metals and contaminants. Again, because they're filter feeders, they have the ability to just take up contaminants that are dissolved in the water and incorporate them into their tissues. So that way, anything that comes to feed on a zebra mussel will then also accumulate that contaminant that the zebra mussel has acquired. So although all of these impacts seem pretty negative, there is potential for positive sort of predator-prey interactions to occur with native predators and the invasive mussels. So one predator that we were particularly interested in looking at this past year was the false map turtle. False map turtles are a state threatened species here in South Dakota, and they are found exclusively in the Missouri River. We were interested in looking at the dynamics between false map turtles and zebra mussels, particularly because false map turtles are known to consume similarly related quagga mussels in other parts of their range outside of South Dakota. And also the related common map turtle has been shown to feed on zebra mussels both in the wild and in laboratory settings. So this led to our research questions. First of all, just will false map turtles consume zebra mussels? We hypothesized that yes, false map turtles will consume zebra mussels. No problem, again, because of that previous evidence of similarly related species consuming them. We also wanted to know if there was going to be a difference in consumption rate between turtles from a heavily invaded area versus a non-invaded area, since we have the unique opportunity to have turtles that have been in contact with zebra mussels for about 5 years now, or about 6 years now, and turtles that have probably never seen a zebra mussel before in their lives. So we hypothesized that there would be a difference. We expected that turtles that have had the potential to interact with zebra mussels prior to this experiment would consume more mussels faster than naive turtles, although we still thought that naive turtles would eventually eat the zebra mussels, just maybe not as many or as quickly as our experienced turtles. So our methods, we collected zebra mussels by hand, just hanging off a dock and pulling them up with my fingers. We brought them back to our laboratory where we measured them lengthwise using digital calipers and separated them into 3 different size classes, small, medium, and large. Then we went out to get our turtles. False map turtles were caught in baited hoop net traps from the 59-mile stretch directly below the Gavins Point Dam, as well as Lake Francis Kase. And of course, we made sure to clean all of our gear and our boat between moving between sites. We didn't want to be the cause of spreading the zebra mussels. So once the turtles were caught, we brought them back to the laboratory where we took standard body data and kept them in individual aquarias. For each trial, 5 zebra mussels of each size class and 5 snails were offered to the turtles. We included 5 snails because we wanted to offer them a native invertebrate prey item along with the zebra mussels to see, like, as a control. And we selected snails again because they're similar to zebra mussels in that they have a shell and they're not as mobile as maybe some other invertebrates are, like chironomid larvae or dragonfly larvae. So after the turtles were presented with the prey items, they were filmed for the first 2 hours of the trial so we could get data on how fast the turtles would interact with the zebra mussels, as well as like the behaviors associated with consuming them, such as handling times. The prey items were left in the turtle enclosures for 24 hours total. After 24 hours, any remaining prey items were removed, and we quantified how many mussels of each size class were not eaten. So going on to our results then, we found that yes, false map turtles will eat zebra mussels. This lovely false map turtle was so excited to eat her zebra mussels that before I even left the room, she was over there chowing down. So I got to have this great video. So you can see how she's chomping on that zebra mussel, trying to break down that shell. That mussel there is pretty big for her to try to swallow whole. So that's why she's trying to mash on it, crunch it into smaller pieces so that she can get to the meaty goodness on the inside without too much shell, also making it down. So it's just fascinating to watch how strong their jaws are to get through those shells as well. Now she uses her claws then to help rip it apart. So fun. So going into more quantitative data, this graph depicts the total number of zebra mussels of all size classes that were consumed after the 24-hour trial, broken down by where the turtles were from. On the left, you can see that in the 59-mile stretch, 45 zebra mussels in total were eaten of all size classes, and only 43 were eaten from the Lake Francis Case turtles. And this really isn't a significant difference, which is pretty cool. So again, showing that yes, false map turtles will eat zebra mussels pretty much no matter where they're from. When we break it down by size class, again, you can see that the 59-mile stretch turtles pretty much always ate more zebra mussels of each size class and more snails than the Francis Case turtles did. The only exception is in the large size class, where about one more large zebra mussel was consumed by the Lake Francis Case turtles. So then we ran an ANOVA looking at the mean number of zebra mussels and snails of each size class that were consumed after 24 hours to see if there were any significant differences between size class consumption. So small zebra mussels were consumed more frequently than the medium and large size classes, and that was a significant difference. Additionally, snails were consumed the most, and that was significantly different from all zebra mussel groups. Part of the reason that we think that is because the snails are more mobile than the zebra mussels themselves. The snails, we noticed, had a propensity to just let go of the aquaria bottom or wall, and so they would just float around with the turtle. That floating movement could have attracted the turtle's attention, and they were more likely to, swing around and eat the snail before finding the zebra mussels just hanging out on the bottom of the aquaria. So again, to summarize our results in terms of our 2 research questions, yes, false map turtles will consume zebra mussels in a laboratory setting. And there was no difference in zebra mussel consumption by our experienced versus our naive turtles. They were about the same amounts of consumption. So some future questions that we have about these interactions. So to what extent are false map turtles consuming zebra mussels in the wild? We're working on answering this right now from data we collected last summer. We collected fecal samples from all of our turtles. So we're still working through about 100 baggies of fecal samples, looking for zebra mussel shells. We know for sure that there was one that contains a whole small size class zebra mussel shell, which was really cool to find. That was a really great day, scoop and poop, but we were really excited to see that. And we also, hopefully this upcoming summer, are going to start looking at if there are any adverse health effects to false map turtles from consuming zebra mussels. I mentioned before that zebra mussels have a propensity to bioaccumulate and biomagnify different contaminants and heavy metals from the water. And so we want to know if consuming large quantities of zebra mussels that do have those contaminants present will have any adverse health effects to turtle immunocompetence or reproductive success. Finally, looking at what are the economic and ecological benefits of false map turtles consuming zebra mussels. We eventually hope to collect more data about if there's any economic benefits, if the turtles are eating enough zebra mussels to potentially be part of any control management strategies in the future. And of course, any ecological benefits, to the turtles specifically. Are they a good source of nutrients with no adverse health effects? And do they provide an abundant food source in areas where false map turtle densities are pretty intense? So lots of exciting work coming up in the future. I can't wait. So with that, I'd like to thank our source of funding for this project was the USD You Discover Summer Scholars Program. The field crew was Danielle Galvin and Vanessa Weigel. These two ladies are absolutely fantastic. Turtle work is hard. Scooping turtle poop is worse. So they did a great job this summer. And finally, some additional support, Bob Garner and the Walls family got us up and running and kept us going throughout the whole summer through the pandemic and everything. So we just really appreciate everything that they do for us. And with that, I would love to take any questions if there's time.
We actually do have about a minute for questions. So if anybody has one for Anna.